Maccario! Luquigi! Maccario! Luquigi! Maccario! Luquigi! Maccario! Luquigi! Maccario! Hey Luquigi, what do you want to do today? I want to learn about the complex topics of genetic manipulation with cross-species applications, so we can make the first jackalope.
Well, I heard that the Guidoidi Biogenetic Laboratory for Genetic Research is the best in the world.
Let's go!
Well, here we are, our local Biogenetic Laboratory for Genetic Research. Do you think we're allowed to go in?
I don't know. I'll go check it out.
I'll wait out here.
Excuse me, can we help you?
I'm looking for the Guidoidi Biolab.
Who are you anyways?
It's me, Luquigi.
I'm sorry, but unauthorized personnel are not allowed in this lab. You need to leave.
How'd it go?
They kicked me out. We need to find a way to get them out of there.
How about a distraction?
Aha!
What was that? I don't know. Maybe just a distraction. Let's go check it out. Okay.
Do you think it worked?
I think I hear them coming. Let's go.
An accident! Let's go!
Let's go in now.
Let's go. What's all this?
Looks like they're notes for their research. They seem to be doing cross-species genetic engineering by putting spider genes into a goat's genome so that its milk will produce the proteins needed for creating spider silk.
According to these notes, there are three types of genetic manipulation: conventional breeding, transgenesis, and cisgenesis. It seems as though conventional breeding is where you cross-cultivate the current organism with a wild relative. This process is repeated using the offspring that exhibit the desired gene. It seems to me as though this is the type of thing that breeders use to make different types of dogs. And cisgenesis is where the desired gene of a wild relative for the organism is placed in the plasmid of an agrobacterium, and then the bacterium is crossed with the current organism. But I don't quite understand this, as it is very similar to a third type of manipulation.
It says the third type of genetic manipulation is transgenesis. This seems pretty complicated, so let's read further. It says that transgenesis is like cisgenesis, but instead of using DNA from the same species of animals, like in cisgenesis, transgenesis mixes the DNA from two different species. It looks like they use a specific class of proteins called restriction enzymes to cut the DNA of one animal in specific locations to separate out a desired gene. These enzymes do this because each one is specifically programmed to move along the DNA and cut only where there is a specific pattern of the four nitrogenous bases: adenine, guanine, cytosine, and thymine. Different types of restriction enzymes will cut the DNA into either a blunt end, in which the enzyme cuts straight through the DNA, or a sticky strand, in which the enzyme cuts through the phosphate and sugar backbone of one strand of the DNA, then cuts along the double helix, breaking the hydrogen bonds, holding the nitrogenous bases together, and then finally cuts through the second backbone of the other DNA strand, so that both sides of the cut have a tail of unpaired bases on them.
So the scientists used a specific enzyme to cut the first animal's DNA at the required location to isolate the wanted gene. They had to experiment with this to find the right type of restriction enzyme. They then put this gene into the genome of the other animal. This was done by inserting the gene into a type of gene construct. A gene construct is usually a small circular piece of DNA called a plasmid agrobacterium. Bacterium DNA is used as it contains natural restriction enzymes that will cut and place the bacterium's DNA containing the desired gene into the host organism's genome. The two pieces of genetic material are finally joined together by a protein called DNA ligase.
Well, I say we have this stuff pretty much mastered. Let's make the first jackalope.
Yippee!
We've done it!
By inserting the gene responsible for antler development in deer into the genome of a rabbit, we've created the first jackalope.
Let's go show the world!